Thanks, I'm Alex. I met some of you already. I just want to say I've had a great time so far at this meeting. It's my first brain function conference. So usually I'm going to MRI conferences or more physiology-oriented conferences. I kind of, I try to see where I fit in here. So, and I think I figured it out. I mean, you guys really focus on the function of the brain, how it works. And I'm a little bit more zoomed out. My research focuses a little bit more on the physiology of the brain. But I hope to convince you today that these things in many ways are extremely related. And that maybe, one avenue that we can think about in the future is how to integrate physiology into function, because I think it's really a modulator of some of the things that you measure. So I have a very general title, mapping cerebrovascular reactivity. That's what I love to do. So I love to study the function of the vessels. So this is one element of physiology that I'm going to try to teach you guys everything I know about it. But first, just to get everybody awake and get me out of my nerves, I want to do sort of like an experiment with you all. And I'm not like a meditation kind of person, but I want to do some breathing exercises together. Okay. So I see someone getting excited. So if I have to do a disclaimer, if you have like anxiety or heart problems or something, then maybe don't do it. But I think everybody here is healthy. So what we're going to do, we're going to try to hyperventilate ourselves a little bit. So we're going to take a really deep breath in. So we'll do one as a practice. So like inhale and then exhale, right? And when I say go, you're going to do this 7 times, okay? And you're going to do it as fast as you can. So in as much as you can, is out as much as you can, and as fast as you can. Ready? So sit up nice and straight. 1, 2, 3, go. So in 1, 2, 3, 4, 5, 6, 7. And now just relax. Wait a few seconds. Anybody feel anything? Put up your hand if you feel something. Anyone wants to say what they feel?
High.
You're feeling high. Okay. So, obviously something happened, right, in our bodies. So what were we doing here? Well, our body's always trying to keep an equilibrium. Like all our systems are searching for this homeostasis or maybe criticality. I don't know what you guys talk about, but something like this. And normally, when we breathe, we're kind of in sync with the metabolism of our body. So the more we work, the more we breathe, because we need to get oxygen into the body and we need to clear the waste product, which is carbon dioxide, out of the body. And what we've done now is we've basically perturbed our system. We've come out of that equilibrium. So we've actually blown out a lot of carbon dioxide, more than what we've been producing. So our arterial carbon dioxide levels have gone down. And the vessels in our body are extremely sensitive to this chemical marker, carbon dioxide. So in your brain, what just happened is that all these vessels, they constricted. So you feel a little bit, dizzy and high, but that's because the blood flow has gone down, right? Okay, now I'll do one more quick one. This time, I want you to, when I say go, just exhale as much as you can, and then just hold your breath, and then hold it. So let's start with put your hand up. Exhale as much as you can, hold your breath, and then just see how long you can keep that. And when you can't do it anymore, put your hand down, right? We're going to see who can do it the longest, okay? So 1, 2, 3, exhale, and hold it. Hold your breath out. Don't breathe. Now we can just be silent for how long it takes. I'll just wait here. Try to fight. Try to do it as long as you can. Three more. Two more.
Katrina, go for a run.
Fight. Hockey player versus the adventurous. Okay, winner. So what did you guys feel towards the end? Uncomfortable, and you I guess you feel like you need to breathe, right? So now we did the opposite. So instead of blowing carbon dioxide out, we kept it in, right? So it was slowly building up, and actually, the interesting thing is your body isn't sensitive to oxygen. So the blood keeps circulating, we keep extracting oxygen, but we're not sensitive to that. What we are sensitive to is the increase in carbon dioxide, right? So you have these receptors that communicate with your centers somewhere, and then this leads to an increased drive to breathe, because again, we want to get air in, we want to blow that carbon dioxide out. So this is kind of the feedback loop. And what was happening in your brain, of course, is that you were your vessels start to dilate. So now we're going the other way. We're increasing our blood flow. So let's say you're in a situation where oxygen is going down, right? Each time the blood circulates through, there's less and less oxygen saturation. So in order to get more, you need to increase the blood flow, right? So this is kind of the mechanisms that are happening. Okay, so that was that. Okay, so now I'm going to start my talk. So we're going to come back to this. But so the goals for my talk today were I wanted to, so I use fMRI, but like, let's say bold imaging, but in a different way. So I thought I will go through a small review of how this contrast mechanism works. I don't know if you guys are, if you have some MRI physics background, I'm not sure. So who doesn't? Maybe who would like to know how the bold contrast? Okay, that's enough for me. Okay, so then I'm going to try to convince you that the structure of our vessels is also very important. And then finally, I'll try to give you some nice examples of what we can do with this information. Okay, so we talked a little bit about blood. So when we breathe in, we set up a concentration gradient, right, in our lungs. We have oxygen-rich air in our lungs, and our body is sort of oxygen poor, right? Because venous blood is coming into our lungs. So there's a concentration gradient, and oxygen flows across that gradient, and it binds to our hemoglobin molecule. So this is my ChatGPT hemoglobin with two oxygens attached. And what's interesting about hemoglobin is that it has this iron center, right? This is what's attracting the oxygen. This is what's binding to the oxygen. And when we have deoxygenated blood, the configuration of this molecule changes, and this iron atom becomes sort of exposed. And what I've tried to do here is showing you when these things are in a magnetic field, that they have an interaction with the magnetic field, right? So arterial blood, as they call it, diamagnetic, so it doesn't really interact with the field, but venous blood is paramagnetic, and it has this effect of sort of concentrating these field lines. And this leads to a local change in the magnetic field. So let's say we're in a 7 Tesla MRI, that's what we use, and you put something in there, depending on on this type of an effect, we call it a susceptibility effect, you locally change the magnetic field, right? So now, okay, I'm going to do another example. So depending on what kind of scans you're doing, you're sensitive to different nuclei. So with imaging, we're looking at hydrogen, right? We're specifically water hydrogen. So imagine this bottle is a hydrogen. Hydrogen is kind of like a little mini magnet, right? So when it's out of the magnetic field, it's just doing whatever it wants. But as soon as you put it in the magnetic field, it aligns with the field lines, right? And the other thing it does, it starts spinning. So it spins with a frequency, which we call omega, and this is proportional to this constant gyromagnetic ratio, and this changes depending on the nuclei and the main magnetic field. So if you have calcium, that gamma is a different value, you have a different frequency. So you need different amplifiers, you need different RF amplifiers, different receive coils to measure those different frequencies, but we're focusing on hydrogen. And now we have this effect, what I just explained before, and this adds a sort of a second term to this equation. So now the frequency is equal to gamma plus the combination of these two things. And you can imagine inside a voxel, you have a lot of these hydrogens. They're all initially spinning in the kind of the Z direction. We give an RF pulse, and then we sort of push it away from that direction, and they start turning like this, right? And this is the signal that we can then measure. If you have a lot of let's say gradients within that voxel, then this frequency will change, and you'll get different, spinning rates, different frequencies. So there's a phase accumulation, and over time, that phase accumulation can lead to a dephasing of the signal, and this is what we call T2 star decay or T2 decay if you're using a different type of sequence. So this is kind of the basis of what we saw yesterday, this QSM susceptibility based imaging. I saw that picture of the Siemens QSM image, so I thought, well, we can also do it on our scanner, so we put a version here. But the interesting thing is what you see is that all these bright spots here, these are all little veins, right? So with these high field scanners, we can go to very high resolutions, and we start to pick up these draining veins. I guess, you can see them like here nicely. And why do we see these? It's because there's a lot of, like as we learned yesterday, there's a lot of iron in there because it's blood, right? And it's veins have deoxygenated blood, so there's this strong susceptibility effect in these veins. Okay, so I tried to give like a toy example. So how this relates to our fMRI signal. So this is, let's imagine we have a voxel, right? And it has an artery in it, right? This artery has, it's just filled with, 100% saturated blood. It has a certain T2 decay then, right, of the tissues, right, because we also have decay just from, local interactions. And we give our RF pulse, we put it into the transverse plane, whatever the signal decays, and at some echo time, we do our EPI readout and we acquire our image, right? This is just a sort of basic fMRI, diagram. So now let's look at a venous vessel, right? So now we have a few more deoxyhemoglobin molecules in there, right? The blood has passed into the tissues, oxygen has been extracted for metabolism, and we have some deoxygenated blood. We have this local dephasing effect, right? These dipoles that form around the iron molecules, and what happens is we get a quicker dephasing rate, right? So our signal decays faster. At the same echo time, we got less signal. Okay, but I'm interested in vascular function. So something that I might do is So just to give you an example of what that looks like, this is sort of a slice through the visual cortex. And you can see here very nicely, these black spots, these are all larger veins. So we have a lot of susceptibility effects there. So you can see very clearly when you do an fMRI scan at this resolution that the signal is basically gone by the time we're doing our readout. Okay, but I'm interested in how the vessels function. So what I like to do, which I'll explain in a little bit, is I want to dilate the vessel. So what happens when you dilate the vessel? I'll go back to kind of the simple brain function analysis, right? In that case, we talk about like neurovascular coupling. So you have an increase in metabolism, which extracts more oxygen, and then a signal is sent to the vessels via the glial cells, right? And the signal is to dilate, right? This is the neurovascular coupling mechanism, and as we probably all know, there's like a mismatch. So the dilation causes an increase in CBF, which is higher than the increase in metabolism. So relatively, you get more oxygenated blood in the veins, right? And this is what we see as an activation. So what I do is similar, but I don't have the activation of the neurons. I just dilate the vessels. So I increase the flow. We push more oxygenated blood through. We get less of this dephasing effect, and then you can see here our signal also goes up, right? So whereas a brain function specialist might look at the fMRI signal as a proxy for neuronal activation, I look at it as a proxy for changes in blood flow and for vessel function, okay? Right, so I have a take-home message, right? So I hope, we all know this probably, but this bold contrast, it's a venous contrast. So it's happening after the neurons, it's happening after the control vessels that actually do the dilation. So it's kind of like we're measuring in the sewer, right? Imagine you have a city, all kinds of things are happening in the city, and you only can measure at the sewer. You know that something's happening in the city, but you don't really know exactly what, right? We just make some assumptions, but it's important to realize that we're measuring in the veins. Okay, so now on to the science part. That was the lesson is now finished. I hope it wasn't too easy for you. So what do we do at the UMC? Well, we do research in vessel function for purely research reasons, but we also have some clinical projects that run where we're interested in, for example, in Moyamoya disease or steno-occlusive disease. How do changes in vascular properties, how do diseases cause these changes? And I personally use this system. It's called a Respiractive system. So it consists of a kind of a computer system with some gas bottles with different oxygen and carbon dioxide blends. And what I can do is I can program in sequences into this system, and it will actually change the arterial oxygen and CO2 values in someone's blood. So what we did at the beginning with our breathing, I can just do that with this machine. You don't have to do anything different. It controls everything, and it can manipulate your blood gases. So this is me hooked up to the system. It's basically a mask. We have to tape it so that it stays sort of airtight. And then in this case, I said, okay, well, just take me to the maximum. So normally you're around 40 millimeters mercury, and here I went up to 65 millimeters mercury. So I guess my blood flow doubled in this kind of moment. And we can monitor everything here. So we can see the person's CO2 levels. We can see their oxygen levels, and the idea is that what we measure at the end expired is equal to what's in the artery. So you can actually normalize your fMRI signal, for example, with the amount of stimulus you gave. So it's a way of really standardizing experiments. It's not very comfortable, so you can imagine what you felt at the end of that breath hold, where you get this increased drive to breathe. You still get that in the MRI, but you're just lying there breathing normally. So sometimes people get a bit freaked out. The success rate really depends on how comfortable you make people feel, because it can be uncomfortable. But I'm good at convincing people, so it works out. Okay, so here's an example this is a patient with brain metastases. So I mean, you can see here the small brain metastases, and you get some edema around these areas. And this is our bold scan taken on a 3T scanner. And then I've just basically looked at the percent signal change now, right? So here I'm giving a CO2 challenge, here I'm giving a CO2 challenge, and here I'm giving an oxygen challenge. So here I'm dilating the vessels, and here I'm just changing the saturation of the blood. And now you can see very nicely that this bold signal, it's lighting up. So the first two blocks, you could say, are representative of the vascular response. And the third one, it's weighted by saturation changes, so it's not a vascular response. So this is more a measure of blood volume, right? So we can look at different parameters using these experiments. And, you can also see that, in these edema regions, we don't see a response, right? So it's just a very simple example of how pathology in the brain can directly translate into vascular function. Okay, so when I started this research during my PhD, which is a long time ago, and I told you guys I liked cycling. Back then I was racing a lot, so I only understood like one speed, which is like full gas, and that's also what I did in my experiments. So we were very curious about what happens at really high CO2 levels. So what I did, I put someone in the scanner. I tell them to hyperventilate, mostly my colleagues, and they called it waterboarding in the scanner, but they still did it. So they would hyperventilate, we would cause some constriction, the bold signal would go down, and then I would progressively increase the CO2 levels to sort of survey the whole range of vascular responses. And why did we do this? Well, we were just curious of whether or not this would saturate, right? So a lot of physiological systems, they work sort of with a sigmoidal response. And if you think about how the vessels respond, they slowly dilate, and at some moment they reach sort of a maximum. And we were just interested, can we pick this up in the scanner, right? And sure enough, as you can see here, I've plotted in one subject the CO2 levels as a function of the bold signal. And how we quantify this is we look at the change in the bold signal per unit change in CO2. So if I use sort of like 5 millimeter blocks, and I see, okay, what was my signal change? That's what this number is. So here you can already see it with your eyes, but as you go to higher CO2 levels, you start to saturate this signal. So why is this interesting? Well, you can see that the 0 point is quite low in this sigmoidal curve, and this is kind of like a control system, right? The vessels are there to maintain homeostasis and regulate in the brain. So this resting point is really around the most sensitive area of this response. So that means that your system is really capable of reacting to quick changes in blood pressure, or I don't know, you go swimming, you hold your breath, all those kind of things. So in a healthy person, this is very, this is where you want to be. But you can imagine that if you have some cerebrovascular disease, let's say you're getting older, your vessels get stiffer, or you have some, blockage in your carotids, and you need to compensate for the reduction in blood flow, then you sort of slowly start to move up this curve. And if you start to move up this curve a little bit too much, then you start to lose the capacity to regulate, right? And you know, from a disease perspective, if you're up here in a certain area of your brain, then you're at a higher risk for stroke, for example. So that's why this kind of things are interesting and important. Okay, so anyways, that was the intermezzo. So we looked at the different tissues, right? So we saw this really nice response in the gray matter. It plateaued, but what we found was interesting is when we looked at the white matter, we didn't see the same sort of plateauing effect. And yeah,
if your pointer is visible at all, it's very subtle. So
let me
really see where you're pointing at.
Yeah. So okay, there we go. So we had this nice plateauing effect. But in the white matter, we didn't see it as much. And you know, you think all vessels are created equal, they should all respond in similar ways. So this was a bit strange to us that this didn't happen. I have to think, slide. Yeah, okay. So we, to sort of characterize, we wanted to quantify things, right? We're scientists. So to characterize this, we fit this kind of sigmoidal response curve. And one of the parameters that you fit is this midpoint. So from a physiological perspective, the midpoint is the most sensitive area of this response. So that was for us interesting. And then what we saw is that in the white matter, this midpoint shifted to the right? So the white matter, you know, it had a sort of a delay, right? There was some kind of dynamics there that we thought. So how did I explain this? Well, if you look at the architecture of the arteries in your brain, they no matter where you go in the brain, there's a sort of common feature is that the gray matter is fed first. So how it works is you have these carotid arteries, they come into the circle of Willis, and then the vessels branch out in the MCA. And you know, from there you have these peel arteries that go over the surface of the brain, and then they penetrate. And from the internal side, you have these lenticulostriate vessels, which come up through the nuclei and then move into the white matter. So you're always seeing gray matter before you see white matter. And we also know that you know, the density in gray matter is at least 2 times higher than in white matter. So there's more blood volume there. There's more vessels that can react to a stimulus. So when we give our CO2 challenge, we start to, you know, evoke a response in these regions. And then eventually we go even higher. The gray matter vessels are reacting. But if I'm like a red blood cell walking along this vessel, and I have the choice to flow to this area, which has very low resistance, or I have a choice to walk along this really long penetrating vessel to these white matter territories, I'm always going to go here first, right? There's going to be a preferential flow towards this gray matter tissue. So this is how we kind of explained it. So as the CO2 goes up, we kind of maximize the response in the gray matter, and at some point the balance of resistances change, and then blood starts moving to the white matter. And this is why we see that the response in white matter is different. So it's driven by architecture, basically, at these high CO2 levels. But again, if you have diseases, these things can also be important. So let's see. So now if I now, so this is an example of our fMRI scan in 70. If I now take a sliding window and I run it through these CO2 values, and as soon as I hit this midpoint, I let the voxel light up. Let's hope this works. This is unfortunate. Okay, well, this video should play. I'm just going to do it like this because it's important. So you see this gray matter coming in first, and then it's following by the white matter, right? So I'll just play it one more time. So I thought this was cool when I first saw this. So this is really showing that there's dynamics and different responses between different tissue types. Okay, so what's the take-home message? Well, the CVR we measure can change depending on the strength of the stimulus, and the arterial architecture introduces some variations in the resistance that can also drive these responses, right? And something that I'll get to later is that tissues can steal blood from one another. So this is kind of an example of that in a healthy situation, but it's way more exaggerated when you have disease, which we'll see later. Okay, so, and just an intermezzo, I mean, you guys are interested in these fine scale, cortical measurements, but these branching patterns, it's kind of like fractal, right? If you go to smaller and smaller scales, you also have these types of branching points. You also have these different control mechanisms. So I wonder if these kind of things can also happen with these more fine scale readings on the cortex. So I have a colleague, Mario, he makes these beautiful sort of vascular models. And if you're interested in, he tries to simulate this stuff. If you guys like models, so if you're interested, I would encourage you to check out some of that work from Mario. Okay, so I talked about dynamics. So I wanted to talk a little bit about how we map dynamics. So there's two ways, right? So that I can, think of right now. We can look at something that we call hemodynamic lag. And to measure hemodynamic lag, what we do is we take sort of an input regressor. So in this case, it's the CO2 reading, this black block, and we just do a cross correlation. So this is kind of like, I guess, when you do connectivity and you look at the direction, it's kind of the same analysis. It's just a cross correlation, and then where this maximum correlation is, that's kind of how we define our temporal dynamics. The problem with this is that it's weighted by the time to peak, and as you can see here, it's insensitive to the onset of the response. So probably the highest correlation is somewhere at the end here, and you know that, okay, well, the bold signal can't go up before my CO2. So it's not really like, it's just kind of like a guessing approach. The second way that I think is more accurate is to look at the signal dispersion. So you could think of this as like a hemodynamic response function, but instead of like convolving it with a neuronal spike, we convolve it with the entire CO2 trace, right? So you can see from the lungs to the brain, there's a lot of things that happen. There's dispersion of the bolus. There's a different response speed of the vessels. So this all gets translated into this kind of vascular hemodynamic response function, and we just sort of fit the time constant that best matches the data. So that's what we call dispersion. We call this tau. Everything is called tau these days, but we map tau across the brain. So here's an example of one subject. So I'm here focusing on the gray matter signal. And what you can see is that the deeper you go in the white matter. The deeper you go in the white matter, the longer this lag becomes, right? So this is a map of the cross-correlation lag. This is a map of the CVR, so just the linear regression of the stimulus with the signal. And then this is a lag-corrected CVR. So what's interesting here is that if you were to use this as a sort of a diagnostic marker, you would see that unless you correct for this dispersion, this delay, you'll underestimate your CVR values. And then dispersion looks very similar to the lag. But what struck me when I noticed this is we looked at the susceptibility weighted images. And you can see that the areas of high lag, of high dispersion, they overlaid with these medullary veins, right? Also the veins I showed on that QSM image. So the question is, are we really measuring arterial responses? Are we really measuring reactivity? Or are we measuring something that's more a consequence of the draining architecture of the white matter? So, I went back into the literature. Japanese people are very good at vascular science. So I found some really nice articles where they explained the organization of the white matter draining system. And basically how it works is you have these sort of, you can think of it like, you have some mountains, right? And it rains over millions of years. You can see all these lines cut into the mountains, and then they converge down into small streams, and eventually you end up in the big river at the middle of the valley, right? So you have these convergence zones, and the white matter is a little bit the same. So you have these areas that collect blood distally, and it sort of pools in through these different convergence zones and ends up in these vessels that are next to the ventricles. And here you can see in this SWI image, these patterns, right? It's kind of like a tree shape. So the question is, if I'm measuring a bold response here somewhere, am I measuring a signal that originated in this area, or is it something that? I gotta go. Sorry. So am I measuring something that started here? Or could it be a response that happened further away And I just only became sensitive to it when enough of that deoxygenated blood aggregated to actually evoke a signal response, right? So this is a little bit how you can think about it. It's also important to note that the venous drainage is sort of separated into different territories, right? So if you look at the cortex, you've got these penetrating arteries, and then you have that, so they go down, and then you have veins that drain back up. So they drain up to the pial surface, and then they go to the sagittal sinus, right? So they kind of go like this, the drainage. But in the white matter, everything goes down. So you have a completely different independent drainage system, and these collect then in straight sinus, and then everything just mixes together at the back of the brain. So, and which you can see here very nicely again in sort of a sagittal view. So I hope this video works now. Okay, so this is how I imagined it, right? So these are little deoxygenated hemoglobin molecules, and they go down through these branches, and then they collect together, right? So there's a huge dissociation between where they were sort of produced and where they end up. And I think that this could be one of the reasons that we see this greater dispersion in the white matter, right? You have increasing blood volume; these vessels get bigger. You have a transit time effect, and then you also have this aggregation of deoxyhemoglobin. So here's an example of what a map, like an exaggerated version of a CVR map, before and after correction for this effect, right? So you see a lot more signal response, which seems to correlate with these large draining veins. So how does this relate to brain function? I've seen papers where people try to talk about, responses in white matter and try to interpret those as like maybe axonal responses that are linked to cortical responses. Well, I don't know if that's true, right? We don't know, because if you believe this, then you could just be seeing a lot of pooling of blood from all different regions, and you don't know if these things are really connected. So that's my caveat for this. Okay, so my take-home messages. So white matter can be underestimated if you don't correct for it. Architecture, again, in the veins is important for modulating this response. And again, just a reminder that bold fMRI, it's a mostly venous contrast. We're not looking at anything to do with the neurons and the arteries per se. We're looking at the output of that, right? Okay. Now moving on to some interesting stuff. So more and more we start to think of the brain as kind of like a holistic combination of different systems. And in the last couple of years, one of the most kind of hot topics has been brain clearance, right? So how do we remove these proteins, these waste products? And when that system breaks down, we get problems like Alzheimer's and dementia, all that kind of stuff. So some work that's been done recently, which in my community was super interesting, is that people have seen that if you, let's say you have a bold imaging slab, right? So that's this green part. It's overlaid on the T1. If you look at this, if you find this voxel here at this point at sort of the fourth ventricle, so this is where the CSF drains into the spinal canal. If you look at this voxel and you look at even your resting state signal, right? If you look at this voxel and you take, let's say you do a bandpass filter between 0 and 0.1 hertz or something like that. You look at these slow oscillations, and you take the derivative of that signal. So you assume that these oscillations are related to blood volume changes. You take the derivative; it gives you the change in blood volume, and you look at this signal. So let's say you do that in the gray matter, and you look at the bold signal response in this voxel. What you'll see is there's a correlation between these two, right? So people have proposed that blood volume changes drive motion of CSF throughout the brain. So we have this really nice data where we don't do resting state, but we have this huge vascular stimulus. So we thought, okay, this is a really easy publication for us, like a super low hanging fruit. Let's correlate the response of our bold signal after CO2 with these signals at the fourth ventricle. So here we've looked at these brain met patients. So this is our paradigm, which I showed earlier. We have this sort of a CO2 challenge with a block and a ramp, and then we have this oxygen block at the back. One second here. This. And here is the gray matter response. So just in healthy gray matter tissue, you can see that the response, the bold response in the gray matter obviously, as you expect, closely follows our paradigms. And we know that this is a vascular response, and we know that this is not a vascular response. Now, when we look at this fourth ventricle signal, what do we see? Well, when these vessels close, we see that, I should also mention this thing called the Monroe-Kelly doctrine. So the brain is this stiff unit, right? It doesn't flex. So anything you bring into the brain, you gotta pull something out, right? So if you constrict the vessels, you reduce the blood volume. And when you reduce the blood volume, something has to come up to replace it. And what ends up happening is that CSF, it sort of gets sucked up in this fourth ventricle and it fills up that extra space. So before we were doing those breathing exercises, we were also driving clearance. We were also making our brains healthier so that you could maybe do this every morning and you won't get Alzheimer's. But okay, so how does this inflow signal work? This is a little bit some MRI physics again. So when we have our imaging slab, we put our RF pulse, we put our magnetization in the transverse plane, and then we have a recovery of our T1, right? We have recovery of the magnetization, and if you wait long enough, it fully recovers. But usually we're scanning quickly, so we only wait maybe 1 second, so we get partial recovery, and then we hit it again. So eventually you reach this steady state where you have sort of a reduced magnetization in your imaging slab. But if you have anything that's outside of that slab, it hasn't seen any RF pulses. So any CSF that we're pulling up into that, through that ventricle, as soon as it gets that first RF pulse, it has full magnetization, so you get a huge signal response. So this effect is quite massive. And you can see it here. This is a 20% bold signal change versus a 2% bold signal change. So you guys have a lot of resting state data. You could try it yourself, and maybe there's some interesting stuff you can find there. So this was our kind of low-hanging fruit. But one thing that we noticed in some of these patients is that, first of all, they had a lot of different amounts of edema, right? So the blood-brain barrier sort of breaks down and water starts swelling up the brain, right? And we noticed that they also had quite a variability in this tau response, right? So somebody with not so much edema seems to have very quick response. So here the blue is the CO2 this time, and the red is the bold response. And then the more edema we had, it seems like this response is getting slower, right? So originally, what we wanted to, our original hypothesis was that in these patients, radiation would damage the vessels, right? So this was the first thing we were trying to learn about, like what's the effect of radiation on vascular response. And so knowing that, you would think, okay, well, probably a more radiated area will have slower responses, right? But we didn't actually see that. What we did see, though, is that areas with more edema had slower responses. So, and I'm not talking about the response in the area, but the sort of healthy tissue around it. So this edema, it has a sort of, it creates a hydrostatic pressure, right? It restricts movement throughout the brain. It's getting a little bit stiffer. And it seems that though this edema is then propagating to the vascular response. Okay, so this was a secondary analysis. So we just correlated the percent of edema with this tau, with this time constant, and we saw this sort of positive correlation. Now, what's interesting is that we scanned some patients more than once. So we scanned them before and after treatment. And in some patients, you see like regression. So a positive treatment response, you see like that edema subsides, right? And in others, the treatment's not working and things just get worse. And they also tend to have more edema at the second time point. And in these orange cases, these were the patients that showed recovery. So when the edema subsided, the vessels responded faster again, right? And in the blue ones, these were the ones that showed progression. So when there was more edema, the vessels responded slower. So what we would have thought initially, like there's damage to this system, it's not working properly. We start to realize now that though the vessels were fine, it's everything else around the vessels that was propagating and affecting their function, because this seemed to be a reversible phenomenon, right? So what does that tell you? Well, basically that the brain mechanical properties are also going to influence the vascular responses or the brain function responses, the physiological responses. So there are some sort of boundary conditions that can be affected when other things in the brain go wrong. Okay, so what have I kind of told you? Well, arterial properties modulate CBR. venous properties modulate CVR, and the brain mechanical properties modulate CVR. So it's getting quite complicated, but it does show that this can be a very interesting biomarker to sort of explore things that happen when things go wrong in the brain. Okay, just to finish now, I want to talk about. I want to go back to this steel physiology that I brought up in the beginning. So let's imagine you have an artery, right? And the artery it's splitting into two perfusion territories. You have some metabolism. Oxygen is being extracted. So in a normal situation, you could consider these two territories equal. You'll have sort of the same saturation in both territories, right? And now when you dilate those vessels, you will have an increase in the CBF, metabolism stays the same, saturation goes up, and you get a bold signal response, right? Positive. Okay, so now imagine you have a situation where through one of these territories, you have like a plaque, or you have an occlusion, or maybe you have some vascular illness that causes stiffening of these vessels. In the case of an occlusion, what happens is that this lumen gets smaller, the blood flow goes down, right? So you have a slower flow to this area for the same oxygen extraction. So your oxygen extraction fraction goes up. So this area in this baseline condition will actually have more signal dephasing than this area here, right? Which is still normal and healthy. So what does the brain do? Well, it wants to compensate for this. So it senses this, sends a signal to these vessels to dilate, right? And then it establishes a normal baseline perfusion again, right? So the interesting thing is, people are walking around on this street, walking by, they've got probably huge diseases and they don't feel it, right? Because their brain is able to compensate for this. Okay, but now let's put one of these people in an MRI scanner and we're going to do a CVR experiment. All right, so we dilate the vessels, and what happens? Well, this tissue can't dilate anymore because it's already reached its maximum, change in resistance. But this healthy tissue, it can. So in this area, the resistance is going to go down, and just like how I explained it before in the gray matter white matter case, a red blood cell walking along this path, it's going to choose the path of least resistance. So blood is going to be shunted towards the healthy regions at the expense of the unhealthy regions, right? So this is a very dangerous situation, and this is how we use CVR to identify areas at risk in patients, right? Okay, so here's an example of one of these patients. So we have in Utrecht we're a Moya center. So patients with Moya around the Netherlands, they come to Utrecht. And Moya is a steno-occlusive disease. It involves a progressive narrowing of large cerebral arteries. And in response, new vessels sprout, but a lot of these vessels are not healthy, and there's a high risk of stroke. So in our protocol, we put these people in the scanner. We run a fMRI scan for about 17 minutes. And after 1 minute, we give them an injection of Diamox, acetazolamide. It's what you take when you go to the Himalayas or something, but this acts as a vasodilator. So it blocks the uptake of CO2. It builds up in the blood, and over about 15 minutes, these vessels will dilate, right? And we look at the response, right? So here you can see on the time of flight, you can clearly see there's sort of a deficiency in vessels on this side. Now over time, this whole perfusion area, you see that it's going blue. So what's happening is that blood is being redirected to the healthy areas where it's going red, and in the blue areas, the blood flow is actually going down, right? So this means that in this person, this whole area is at risk of stroke. And just to give you an interesting story, we once had a young child, because it's predominantly in children, and they were playing the flute, right? So when you play the flute, you have to blow a lot, you have to breathe in a lot, just like we were doing at the beginning of today. And what happened was that because of that increased ventilation, the vessels constricted, and she got a ischemic attack, right? So that was the reason that child was brought to the hospital, and then later you find out that they have moya using one of these kind of scans, right? So this is how we start to use this knowledge in the clinical practice. And here's another example. So on the right, this is a dynamic angiogram. I fit a model to it so I can sort of see the inflow of blood through the large vessels. So this is also a patient with moya. And generally, when you see that the hemodynamics is severely impaired, the neurosurgeon looks at these images and says, okay, well, we need to do a bypass surgery. So with a bypass, you have a direct or indirect bypass. So either you take a vessel from outside the brain, you make a hole in the skull, and you kind of connect it, right? So you get a collateral flow. Or you take a hole in the skull and you take one of these muscles. I didn't even know this existed. You take muscle from like a flap of muscle and you put it on the surface of the brain, and then vessels kind of grow into the brain. So that's an indirect versus direct bypass. So here you sort of see these bypass vessels, so direct bypass. And you can sort of see a delayed arrival of blood in this sort of, if you look with squinted eyes. But if you look at the bold now, so this is the same scan, and I just show this because it's interesting, obviously. But if you look in this corner here, what do you see? Like first you see it goes blue, and then it goes red, right? So this is a very, to me, interesting dynamic because this bypass vessel, it's a large vessel, but it's completely outside of the normal path of feeding arteries, right? It's kind of artificial. And it has its own resistance properties. So when the rest of the brain is kind of dilating, the blood is going preferentially to those regions inside the brain at the expense of this bypass. So first you see this blue region, but then eventually it catches up again, right? And to me, this would be a successful, successful bypass, right? Because in the end, it's getting the same reactivity as the rest of the brain. It's just delayed a little bit. It's delayed a little bit, and there's a different resistance dynamics involved there. So if it wasn't working, you might expect this to stay blue, for example, right? Like what we saw in the previous example. So by combining these different physiological MRI methods, we can start to interpret the disease state in these patients a little bit more, finely or whatever. Okay. All right. So take-home message. Steel physiology manifests as negative bold signals, and in my world, in the physiological world, this is a bad thing, right? This means that the system is not working the way it should. Sometimes I wonder if I've read papers on activation studies, and people talk about negative signals in the same region as positive signals, and they try to attribute this to inhibition. And yeah, I always wonder, is that the case, or are we just maybe seeing local steal effects where some regions are activating and then locally these small vessels are still branching? Maybe some of that blood is just being redirected, and we see that as a negative activation, but it could just be a flow effect. But that's just sort of something to think about. Okay, so a pitch for this kind of data. I have this website. If you're interested in this stuff, you can check it out, and you can try some of these pipelines and make your own CPR maps. Interestingly, you can also do this with resting state data, right? If you assume that these fluctuations are really vascular in origin and not neuronal in origin, there are ways to sort of infer reactivity, or at least blood volume. So we have some of that stuff on there as well. And with that, I want to thank you guys for the great time I had in Sweden. I have to go soon, but if you find this interesting, you can always email me. I'm happy to talk about it, and if you have any questions.